All right, let's move into module 2.1. Let's talk a little bit about this plant disease cycle as we set this up, as we go into some of the diseases that we might encounter in crop production. And so I might not have gone over sort of any of the insects that we're going to encounter in crop production, because I want to do that at the end when we talk about like integrated pest management. So in this particular module 2.1, we'll at least introduce how diseases start and also some of the diseases that you might encounter. And then we'll combine that all the way into module 3, where we're talking about insects and different fungicides like insecticides and fungicides that you might use. Okay, so in order for a disease to exist, we need to have 3 things. First thing is a host. There needs to be a plant there, or else the disease has nothing to feed on or extract energy from. Like, that is kind of the purpose of insects and diseases. Well, not the purpose, but that is like why they're a pest, because they are seeking energy from the same thing that we are trying to grow for profit. And so we need to minimize that. So first thing we need is a host. Okay? The next is going to be the pathogen. If the pathogen doesn't exist, then we don't have a problem. And then finally, the environment. And if the environment is not conducive, then you probably don't have any of these, because let's say that it's, in November. We're probably not growing much corn in November. Also, the temperature is probably not conducive for the disease to be growing. Maybe the pathogen doesn't even exist in that environment. Now, however, there could be some differences for, let's say, maybe turf grass. There are some turf grass diseases that thrive in a cooler environment. So having an understanding of what the environment is, am I susceptible to having this infection by this pathogen based on my environment? So we need a host, we need a pathogen, and we need an environment. And you could also say that we need the same thing for our insects as well. And so some of the properties that we're going to have are like, the level of virulence, and virulence just being like, like how strong is it? So many of you remember from COVID, it might be that you had COVID, but you were asymptomatic. And so, maybe it wasn't that you were hit that hard, or maybe you knew someone that maybe, didn't go down as bad. And so we can have different levels of virulence, like how strong is that pathogen? Can that pathogen adapt? Like, what is the adaptability of that pathogen? Can it withstand insecticides? Fungicides? Can it change based on plants? Can it grow in environments that might not be absolutely perfect for it? Kind of like facultative or obligative. How well is it dispersed? How long can it survive? And then can it reproduce quickly? And so some of the things of the host that are going to influence that are how healthy is the plant. And so if the plant is healthy, there are some diseases that the plant can just outgrow. It's kind of similar to when we get sick. If our immune system is strong enough, then we can outlast that infection. But if it's not, if we have an unhealthy plant, then it is likely to be more susceptible to those attacks by those pathogens or by those insects. And so, like, why would you not want to go? Well, it's already dying. We'll go ahead and finish this out, because we know that it is a source of energy that is not going to give us a problem. What the growth stage is and what the form is. And so it might not be that, the plant is big enough yet in order for it to fight off that insect or that pathogen. And so it might be that in the early seedling stage, if you don't have a healthy plant, then you might not have a stand, especially on like things like turf grass. We tend to not have many problems with like row crops as far as like in early stages. Then that's because the environment might not be conducive for that host yet. Population density and structure. So this one is going to be important for crop production specifically, in that think about corn or soybean, in that if you have high populations, is there going to be like, I want you to think about being underneath in the row of a corn plant, like in a cornfield. Like, what do you think that environment is? Do you think it's hot, or is it cool? Well, it could be a little bit of both. If we have enough heat trapped in there, like it's probably not, the soil temperatures might be cool, but the air might not be cool. Do you think that it is humid? And why do you think that it might be humid? What are some of the reasons why it might be humid? What about soil moisture? And um, we we have some evapotranspiration going on. Is that moisture going to be able to make it out of that canopy? What about airflow? You think there's a lot of airflow underneath in between the rows of corn? No, probably not any of those, right? So we could probably assume that in general, not always, that if we have very high populations of crops, elevated populations, then there is likely to be a hot, humid, shaded environment that is really very conducive for these pathogens or even these insects, and we might not even be able to see them. We might not even be able to recognize that. So, have an understanding of like population densities and that you don't want to overpopulate your fields because you're kind of influencing the airflow, and we'll find especially with fungi and at least with fungi, that we really need some airflow to move through that. That's really what's going to influence how much that pathogen survives. I also wanted to come back here and address for vegetable production. And so vegetable production will be a little bit different in that those pathogens are likely to be more soil borne, and they are likely to feed on like root systems, and that is why we might not have a healthy plant that can outgrow that. So in our crops, in our row crops, we probably don't have an issue in the early stages. We have an issue in our later growth stages when that plant is larger. Whereas in vegetable production, we might need to be looking at early growth stage potential for disease infection. So properties of the environment, again, the temperature, how much rainfall have we had? Are the leaves wet often? What soil properties are influencing that, and wind, as I just mentioned. So let's go back to the corn canopy. Do you think that it is hot or cool underneath those corn plants? What about the rainfall, or what kind of dew? How long does it stay wet? If it is, let's think about that. If you have a clay soil, would you expect that soil to stay wet longer than a sandy soil? And what happens if you have a lot of water standing on your soil with your roots? That's going to go bad for a lot of things, isn't it? So here we bring in soil properties in that we need some proper drainage to go on there. And then finally wind, and wind is actually going to affect all of the ones above, because if you have enough airflow through there, well then you're likely to move some of that water off of that soil. You're likely to reduce the leaf wetness period. If you have wind moving through, you don't have to wait until 1 or 2 o'clock in the afternoon in order for the dew to come off of your corn plants. And it might change the temperature. For those of you that are managing turf, we have this also, in that we're going to apply water early in the morning because we kind of want to move some of that dew off, because we don't want to have extended periods of leaf wetness. And we might not get a lot of wind over our greens because we have trees that are blocking the way. So there are some management things that you can do as well in order to minimize like just the potential for having these problems. Okay, so you probably need to put a big star on this. Many of you have had me before. Generally, I have these notes with open spots, but I didn't have this one in particular as I've been going through and trying to recall some of the content that I had from fall of 2022. Okay, so put a big star by this. You're going to see this one again, and this is important because these are the stages of disease development. So first we have inoculation. And inoculation is going to be when that pathogen arrives on that host. So this is a spore that falls on a plant, and then once that spore falls on that plant, it begins to attack it. Part of that attack is penetration, and we'll talk about some of the ways that those fungi actually penetrate into, or get into our plants. Next, we have infection. And so once the inoculant has landed on the plant and begins to penetrate, once it makes its way in, it starts to infect it. And when we see this in a couple of slides, it's going to make sense. Next, once it's in there, it will start to grow and reproduce. Remember, it's trying to find energy in order to preserve its life. And then finally, we have dissemination. So once it's infected the plant, it can then, we have a lot of these different spores. How do those spores then continue their life cycle and disseminate through our crops and pretty much decimate it if you don't catch it in time? So, inoculation, penetration, infection, growth and reproduction, and then finally dissemination. And this one's going to be important because like the dissemination, we want to make sure that we're not tracking this. And I know for certain, if you work in a greenhouse or if you work on like turf grass, you can track disease all across your whole golf course and not even know it. Okay, so inoculation, that's the arrival of that pathogen on the host, and then we have the inoculum. And the inoculum is the actual spore, right? And so we can have primary and secondary based on how they cause the infection. So you get a spore that lands on a leaf, that spore penetrates that leaf, that is the primary inoculum. During the growth and reproduction, that is the secondary inoculum. So you can think about this as the mom or the dad, and then this being the second generation of that inoculum. So wind, water, and insects are going to be the primary way that we have these pathogens carried. Insects are vectors, and so that's why they can be an issue. So not only are they feeding on our plants, but they're also carrying this inoculum that can infect our plants, and kind of a combination of not only are we killing the plant with the insects because they're taking the energy, but we're also getting inside of the cells and we're destroying it from the inside out. So we have, the germination of these spores, maybe in fungi. We have, attachment, so virus and bacteria, and then nematode eggs. And we probably don't have much of a problem with nematodes in central Tennessee, like in the upper Cumberland, because most of our soils are clay soils. If you remember back from soils, when we talked about those different organisms, nematodes were one of those that like to feed on the roots. So maybe out in West Tennessee, where they have more sandy soils, they probably have a bigger problem with nematodes, because think about the pore space. In a clay soil, probably not as much pore space, but in a sandy soil, those nematodes are probably freer to move around. So it's really a combination of all of these things. So the most common type of, at least penetration are going to be by fungi and nematodes. Nematodes have this stylet that actually like a needle that penetrates that host. And then once that's in there, it begins to remove the nutrients that it needs. And then if it has some soil-borne pathogens as it's crawling through the soil, when it injects it into that plant root, it's also infecting the root with maybe with the fungi, or maybe with a bacteria or a virus. So nematodes have, we got insects doing it from the top, from the top of the plant, and we have nematodes catching it from underneath. We've got insects that are possibly feeding on these. It's just a big energy system, and having an understanding of the environment, what potential pathogens that we can have are going to help us minimize the damage done by crop pests and disease. All right, big star on this one, because there are three ways that we can invade a host. We can do it directly, in that As we land on top of that leaf, you might not be able to see it here very well, maybe in your notes or if you've downloaded the slides. We have this spore that is on top of this leaf, and it moves through those plant cells and gets inside of that plant, and the mesophyll cells starts to penetrate. We get growth and we get infection, and then we get growth and reproduction through natural openings, the stomata, the cell. And so remember that our stomata are going to need to open up to allow gas, or maybe even allow some water to move out. We have eutation, in that we have too much water in the plant and has to get rid of some of that. Maybe you've seen this. Or through a wound. Insect comes in, fall armyworm comes in and starts chewing on a plant. That gives a direct opening for this spore to just move straight into the cell. It doesn't have to spend any energy getting into this, making its way through the cuticle, trying to chew that. We have the infection, and then all of a sudden we just basically have cell disintegration. This now opens it up for more spores to come in, more insects to be able to chew on this and get their energy as well. So again, with the direct penetration, we're going to have some mechanical force that softens that cell wall. They may have enzymes. Maybe they can just grow straight through that. Then we have our aprosorium and the penetration peg. And so once that penetration peg makes it in, we've now been through this cuticle and the cell wall. And once we're inside the cell wall, it's like breaking into a safe, right? Once we're in, the disease is doing what it's doing. So I want you to think about this as we move into module 3 later on down the road, that we have, much like our insecticides as well and our herbicides, in that we have contact and systemic. And so if you know that you're going to have this outbreak, you have the potential for an outbreak, you'll want to apply a contact fungicide, and that when that spore lands on that leaf, it starts to attack it. If it's already made it into the plant cells, then you might need to apply a systemic fungicide, or bactericide, virucide, whichever, in order to minimize the damage of that disease. Okay, 3 things for direct penetration, big star here, the aprosorium. And the aprosorium is kind of like, it is the base. It's where we start attaching to that. Kind of like if you've ever seen a backhoe or a large piece of heavy equipment that's going to do some drilling. That's got the feet that come down that kind of give it that strong base so that you're not just moving all over the place. That is the aprosorium. We are connected here, and we are going to start drilling or penetrating with the penetration peg. And then once that penetration peg makes it in, we then have our intracellular mycelium. And so once, that just kind of grows in between those cells, right? Actually, this would be intracellular. This is going to be into. Intercellular is between. This is the intercellular. This would be intracellular. This is where we're getting the energy from, right? And once we infect the cells, we're going to, the virus, the fungi, whatever it is going to replicate. It's going to multiply based on the energy. Once it kills one cell, it kills another, it kills another. Now we've got this large population of this fungi that is just going to eat everything up. And the plant doesn't have any defenses against that, because you're killing all of its soldiers or its cells in order to fight off this infection. No different than when we get infected. So our natural openings are going to be the stoma, the stomata, as I mentioned. We've got hydatodes and lenticles, and so these are some of the intercellular spaces. As you can see, we've got this spore. It has now made it into some gap in our leaf, and is starting to intercellular, and once it makes it intercellular, it will put the intracellular mycelium and begin to attack all of these cells. So it kind of engulfs it and then extracts all of the energy. So the three natural openings are going to be the stoma, the lenticle, and these hydathos. And hydathos is just that there are these places on our plants that can release excess water. All right, here's another look. These are our lenticles. These little extra bumps on our bark. We've got the stoma, and then you can see along the leaf edges, here is our hydathode. And so it is possible, like we need to understand that most fungi move through water, and if you get in the water, then you have a direct route into the opening of that plant. All right, so once we're in the plant, it's gonna start infecting it. That fungi is gonna start doing what it's supposed to do. It is gonna start attacking those cells and those tissues because remember, it's trying to obtain the nutrients from them. And so while we don't necessarily think that there's a lot of nutrients in a cell, when we talk about organelles and whatnot, but there are a small amount. Remember, these are microscopic organisms, so it's not like they need pounds or parts per million per acre. They just need a little bit of energy so that they can replicate. And so once you get infected, there are going to be symptoms. No different than a cold. When you get a cold, there are symptoms. What are some of those symptoms? Runny nose, fever, hot flashes, might be cold flashes, could be coughing. Something is different. You're not fully healthy and you can tell that. And so the symptoms and the signs, we'll talk about those when we get into actually looking at the different diseases. The symptoms and the signs are important. A symptom is something that you notice that goes, this isn't right. Something looks wrong with that plant. The sign is when you actually pull a leaf off and you can see the spore. It's like the verification. So we'll have an incubation period and then also this latent infection. And so how long before that plant is inoculated, the spore lands, that is the inoculum, we get inoculation, penetration, infection, and then we see the symptoms. Because there is going to be this latent time while you don't know, much like none of us know the exact time when we got infected with a cold. So that cold, that whatever is happening inside of our bodies, takes a minute for it to incubate. And then we start to feel the symptoms, and then we go to the doctor and they say, you have strep, COVID, the flu, whatever it is that you might have. And so we have the same thing with our infections. And this goes a little bit different than when we talk about like economic entry level and economic threshold for insects, because we don't know until it's already happened. Like for humans, we can drink orange juice all we want to. But if you don't drink orange juice, when should you have started drinking orange juice in order to beat your immune system, like in order to beat that up before you got infected? But you don't know when you got infected. So disease management is a lot different from insect management. All right, so growth and reproduction, we have four main biotic factors that are going to influence this. Start right here as well. We've got fungi, bacteria, viruses, and nematodes. And so how these reproduce are important for what type of chemical we're going to use for that. And so for fungi, we want to make sure that we have a contact fungicide, at least on the outside to start with, so that if the fungus lands on the plant, there is some fungicide that is there to kill it before it starts its thing. And so much like If you're going to spray a contact insecticide, like let's say you go to like 7. Let's say you go to Lowe's and get some 7 and sprinkle that on your plants. When the insect lands on the plant, it's getting the 7 on it, and it should kill it before it causes any major damage. The bacteria side is likely going to be targeting some sort of cell division mechanism. Viruses are going to be a lot different. Once you get a virus for plants, it is very difficult to get rid of that virus because it is replicated by the cell. And once it has made it into the cell, that virus has attacked that cell, it's actually in the DNA. And so once it's in the DNA, that is going to be in your plant forever. What if it's in the soil? That's going to be in the soil forever. You cannot get rid of it, or it's very difficult to get rid of. Like you almost have to, you basically have to sterilize your entire soil. And on 100 acres, sterilizing the soil really is a last ditch effort. You really don't want to take it to those measures because you're using things like methyl bromide, and that's very toxic to the environment. You got to put a tarp out, and I guess this is for like my turf students. Maybe you've seen this or at least heard about it. They put a tarp out. You come in and you spray this toxic, basically like the bug bombs, but you're putting it under a tarp and you're allowing that to seep into the soil, and it basically kills anything biotic, anything alive in that soil. And so let's think about what that does to all the good bacteria, all the good fungi that are in your soil. Is that a positive thing or a negative thing? Yeah, it's challenging, isn't it? Because you have to pick the lesser of two evils. And so this is where having an understanding that like methyl bromide is a last issue, as like a last resort, because you're going to kill everything in that soil. How do you replenish it with the good fungi and the good bacteria? Probably not any good viruses or nematodes. Okay? Next, how do they get disseminated? You've heard me mention this before. It is movement with water or wind. Wind should be in here as well. If you have the spores on a dry plant, the wind can pick that up and carry it as well. But for pathogens in the soil, rain or irrigation water that move on the surface, and so you have, water that is in the soil, you have water that is on top of the soil, you have a pathogen in the soil that is moving through water. It just loves moving through water. And if you have a pathogen in the soil that moves up through that water and gets on the surface runoff, that can then be transported. And for my turf people, if you have a sloped fairway and you have water running down that sloped fairway with fungi in it that is going to kill your turf grass, then that's not going to look pretty, is it? If you're in row crops and you have a lot of runoff, you might not even know that you've got a pathogen problem because you can't see it until after the symptoms show up. You don't even know. And then, as I mentioned, insects and nematodes and other vectors. And so when I say insects, I want you to be aware that this is not only beneficial, or non-beneficial, right? There's not only pests, but it's also pollinators. Beneficial insects can also carry this. Like bees don't know they're picking up the spores that are on a plant. And then nematodes are going to be bad because they have that stylet, and if they have the vector, if they have the virus inside of them, when they put their nose or their stylet inside of that cell, they're directly injecting that virus into that cell. And once that virus gets into the cell, then it becomes part of the DNA. Once it becomes part of the DNA, it's very, very difficult in order to fix that. So having an understanding about what is actually moving the pathogen can save you a lot of trouble down the road. What about transplanting or seeds? This might be for my greenhouse workers. Let's say that you want to take some, you see a plant out in the field that you're like, I would really like to have that in the greenhouse, and I want to propagate that, and I want to continue growing a population of those, but you don't know if it has a disease on it or not. Now you brought it into your greenhouse, and what is the environment in a greenhouse? Is it hot? Is it humid? Is there a lot of air movement? Are plants in close relation to each other? Now we're starting to see like just how bad that can be. It's possible that you use tools outside and then you bring those tools and use them in the greenhouse. So I'm not sure if Dr. Earhart or Dr. Pierce talk about this, but making sure your tools are clean before you like mess with different crops. Clean your tools before you bring them into the greenhouse. It could be that, they didn't even know that they had a pathogen and it was sold at, Johnson's Nursery, and you put it in the back of the truck for a day or, whatever. They didn't know they had it. And in 3 days you bring it back and now all of a sudden your stuff is now infected. And then humans. It could be that we have, we've, through handling of the plants, whether you have gloves on or not, if you're messing with a disease-infected plant outside at Shipley with gloves on, and then you bring those gloves back into the greenhouse and start messing with plants in the greenhouse, you're a vector. Our tools, we need to make sure that we're cleaning our tools before we bring them back into a greenhouse. And that's a little challenging out at Shipley. Again, I'm not sure how much Dr. Earhart or Dr. Pierce have talked about that, but I guarantee you that if you go to a large greenhouse, like maybe you go up to Tanamuri and Antley up in on the way up to Livingston, or you go to Johnson's Nursery and they're doing some work around there, they probably have they probably have strict cleaning rules because they don't want to bring that into their greenhouse. They've worked hard for that. Contaminated soil, what if a container is contaminated? You got infected seed or infected nursery stock and you didn't even know. And now you've got a whole entire infected greenhouse and you have to start all over from scratch. So to finish all this up, it's important to understand how this happens and how to stop it before it becomes a problem. Insects, we can kind of do a little bit about that. We have sticky traps and we can see when populations start to increase, but diseases, man, it could be a while and it could pop up, and then we have some turfgrass diseases that like turf and vegetable pythium. You get pythium either on your turfgrass or you get it in your vegetables and you didn't find that in time, 2 or 3 days and you're done. You don't even have the time to spray a fungicide. It's already infected and took it over. And there's nothing you can do about that. So just want to make sure that we start at least to begin thinking about the process of getting an infection, how it moves, and how we can stop that with our, the way that we movement with water. Are we making sure that we're draining properly? Our insects, as we discussed in Module 1, we need to make sure that we're monitoring our populations. We could get some transplanting, and then it's mostly going to be humans through, just handling of diseased plants. They may or may not have even known, but it pops up, and then all your crops gone. All right, so that's going to end it for Module 2.1. The quiz should be available. It's just a simple quiz. I know I didn't have the blanks in there like I normally do, but I also did tell you where what to star, and of course they're open now. All right, we'll see you on Module 2.2.